distribution. Okay, last class completed dicot stem, internal anatomy, and leaf anatomy. Okay. Okay. Stomata. Okay. First distribution. Okay. Distribution of stomata on the basis of stomatal distribution. Okay. Three types or three categories. First one is amphistoma, hypostomata, and epistomata. Amphistomatic, hypostomatic, epistomatic. Okay. Amphi means both. Stomata present on both surface of leaf. Okay. That is amphistomatic condition. Okay. For example, most of the herbs shows both surface. Okay. Stomata present on both surface. That is amphistomatic. Okay. And second one is hypostomatic. Okay. Hypostomatic. Hypostomatic means stomata present on lower surface of leaf. Okay. Stomata present on lower surface of leaf. For example, most of the trees. Next, epistomatic. Okay. Epistomatic condition. Stomata present on the upper surface of the leaf or adaxial. Okay. For example, nymphia. Okay. Leaf shows two surface or both surface, means upper surface, lower surface. Upper surface adaxial. Another name is adaxial, and lower surface is abaxial. Abaxial surface and adaxial surface. Okay. Stomata contain three parts. Okay. God cells, subsidiary cells, and epidermal cells. Okay. That is called stomatal complex. Stomatal complex. And dorsiventral leaves. Okay. Anatomical classification of leaves, dorsiventral. Dorsiventral leaves with distinct upper and lower surface. Dorsiventral leaves, distinct upper and lower surface. Okay. Commonly called upper surface adaxial, lower surface abaxial. Okay. These occur in dicots. Okay. Dicots occur in dicots plants. For example, mango, banyan, nerium. Okay. Mango, banyans, and neriums. Okay. Next, unifacial leaves also there. Unifacial. Okay. Leaves are cylindrical structure. Okay. Cylindrical structure distinct into upper and lower surface. Cylindrical structure and shows upper and lower surface. Okay. Is absent. Upper and lower surface is absent. These occur in species of Allium. Okay. Allium shows unifacial leaves. Okay. The main character is upper and lower surface is absent. Okay. Leaves are which type of structure shows? Cylindrical structure. Okay, students. Which structure? Hello? Cylindrical structure. That is the information, the complete information of stomata. Okay, stomata and the stomatal conditions. Next, types. Okay, stomatal, stomatal types. Okay, commonly stomatal types we are observing four types. Okay, four types. That is structural types. Okay, generally, God cell shows kidney shape in dicotyledons. Okay, kidney shapes. Okay, in monocotyledons, uh, God cell shows double dumbbell. Okay, double dumbbell. So this is the main difference between monocot and dicot God cell structure. Okay, dicot God cell shows kidney shape, kidney shape cells, and monocot shows double dumbbell. Okay, students, double dumbbell. Okay,
So what's the page number?
what?
Page number.
Page number 37. Okay, page number 37. Okay, here stomata is a minute epidermal opening with the following characters. Okay, that is shows mainly kidney shapes. Next, types. Okay, anamocytic, anamocytic, anisocytic, parasitic, diacytic. Okay, first observing one leaf disc. When we are observing leaf disc, one small leaf disc. See here, one leaf disc shows number of stomatas. Okay, see here. Okay, leaf is ficus. Here, see the leaf is ficus tree leaf. Okay, we are taking only small spacious disc, that is called leaf disc. One leaf disc shows number of number of stomatas. Okay, stomata contain different parts, that is four: God cell, subsidiary cells, epidermal cells, commonly called stomatal complex. Stomatal complex. Okay, the structural type is first one is anamocytic or irregular. Anamocytic or irregular. Okay, anamocytic or irregular means, okay, stomatas are four basic types. Okay, four basic types. First one is anamocytic or irregular or ranunculus type. Ranunculus type. First, anamocytic means, okay, anamocytic means surrounding epidermal cells have, okay, anamocytic means surrounding epidermal cells have no special arrangement. Okay, no special arrangements. They are all similar. There are no subsidiary cells. Okay, all cells are normal epidermal cells without any modification. Okay, stomata surrounded by epidermal cells, all are normal condition. Okay, the type of stomata is called anamocytic. Okay, anamocytic. See here, this is anamocytic stomata. Okay, anamocytic stomata. Okay, next, second one is uh, anamocytic. First one example: Anomocytic example, Tridax procumbens. Okay, Tridax procumbens shows anomocytic stomata. Tridax procumbens. Next, Borehavia diffusa. Borehavia diffusa and Cleome species. Okay, all these plants are shows in their leaves anomocytic stomata. Okay, stomata can surrounding subsidiary cells. Okay, all are normal. They are not shows any modification, any special structure, common structure, that is anomocytic. Okay, here see here anomocytic stomata surrounded by epidermal cells, subsidiary cells. Okay, this is anomocytic. Next, second one is anisocytic. Okay, anisocytic stomata surrounded by three or more subsidiary cells. Okay, one of them is smaller that the other like. Okay, one of the smaller, remaining are larger. That is the type of condition is aniso. Okay, anisocytic. Anisocytic means unequal. See here. Okay, centrally arranged God cells surrounded by subsidiary cells. In subsidiary cells, one is small. Okay, remaining subsidiary cells are big size. This type of arrangement, okay, is called aniso. Aniso means unequal arrangement. That is called anisocytic stomata. Okay, or cruciferous type. Another name is cruciferous type stomata. Okay, cruciferous type stomata. Okay, um, this type of stomatas are we are commonly observing or found in Malvaceae families. Okay, Malvaceae families and Brassicaceae family plant shows anisocytic. Okay, next parasitic. Okay, parasitic stomata. Okay, stomata with two subsidiary cells. Two subsidiary cells arranged in parallel. Arranged in parallels. Okay, with cojoint walls. See here, cojoint walls at the pole. Okay, at the pole. This type of stomata is called parasitic. Okay, parasitic are parallel celled stomata. Parallel celled stomata. Okay, another name is rubiaceous. Parasitic or rubiaceous. Okay, subsidiary cells are arranged parallelly. Okay, to the god cell. Okay, that is parasitic stomata. Parasitic stomatas are found in rubiaceous family. Rubiaceae family. Hypomia palmata. Okay, rubiaceous family and Hypomia palmata also shows parasitic stomata. Okay, fourth one is diacytic or crass wall. Okay, diacytic or cross-walled. Okay, stomata with two subsidiary cells here. Okay, polar in position with conjoint walls at a right angle to the guard cell. Okay, at right angle to the guard cell. See here, two subsidiary cells. Okay, polar in position with cojoint walls, right angle to the guard cell. Okay, right angle to the guard cell. This type of stomata is called caryophyllaceous. Caryophyllaceous stomata. Caryophyllaceous stomata. Commonly found in Acanthaceae. Caryophyllaceae. Many Lamiaceae families shows this type of diacytic or cross-walled cells. Okay, and also peppermint, mentha, piper. Okay, all are shows diacytic stomata. Diacytic stomata. Okay, this is the important topic of leaf stomata. Okay, stomata and stomatal structure. Stomata and stomatal structure. See here, all are there here. Okay, stomatal structure. Okay, anamocytic, anisocytic. Okay, next parasitic, diacytic. Okay, actinocytic also there. Number of epidermal cells surrounded by God cell. Okay, this is actinocytic. Okay, all these are the types of stomata. Types of stomata. Next, okay, types of stomata. Example already discussed. Okay, parasitic or rubiaceous. Okay, coca leaves, senna leaves, diacetic, peppermint. Okay, already discussed. Next topic. Next topic is uh, secondary growth. Okay, already we are discussing about anatomy of stem. Okay, stem and root primary structure and anatomy of stem and root primary structure over. Next, anatomy of different plants stem. Okay, stem anatomy. That is before going to the anatomy of stem, secondary growth. Okay, secondary growth. That is the block 2 topic. Block 2 topic, secondary growth. Okay. Okay, secondary growth. The most of gymnosperms, most of the gymnosperms shows this type of secondary growth. Okay, secondary growth. Gymnosperms and dicotyledons shows increasing the thickness of the stem, increasing the thickness of stem and root by activity of vascular cambium or cork cambium. Okay. The definition, the increasing in plant growth due to vascular cambium or cork cambium is called secondary growth. Is called secondary growth. Okay, increase in plant growth. Increase in plant growth due to vascular cambium. Due to vascular cambium or cork cambium is called secondary growth. Okay, is called secondary growth. Whereas the growth that occur due to abnormal position and abnormal activity of cambium is called anomalous. Anomalous secondary growth. Anomalous secondary growth. Okay. Anomalous secondary growth generally two types. Okay. First, uh, first type is vascular tissue or formation of cambium. Formation of cambium. Second one, protective tissue. Protective tissue. Okay, vascular tissue means xylem, phloem. Okay, xylem and phloem developed by vascular cambium. Developed by vascular cambium. Okay. Next, the formation. First, normal secondary growth. Okay, normal secondary growth. Increasing in length. Okay, increasing in length of the shoot and root is referred as primary growth. Is referred as primary growth. Okay, it is the result of cell division in the shoot apical meristem. Okay, all these activities shows shoot apical meristem. Shoot apical meristem. Okay, that is the main important tissue is shoot apical meristem. The process of secondary growth can be divided into two parts, that is intra secondary growth and extra stellar secondary growth. Okay, intrastellar secondary growth, this process takes place in the stellar region. Stellar region means xylem and phloem region. Okay, first formation of cambial ring. Okay, formation of cambial ring. The young stem vascular bundles are limited. Young stem vascular bundles are limited. Collateral open type. Okay, collateral and open type vascular bundles. Okay, these bundles shows strips of cambium between xylem and phloem. The center part is called fascicular cambium. See the ring? Okay, one ring is observed at the point of xylem and phloem in between. That is fascicular cambium. Hello, students. Hello.
Yes, sir.
Fascicular cambium is formed at the center position of xylem and phloem. That is fascicular cambium. This fascicular cambium play major key role of secondary growth. Okay, giving results, increasing the girth of the stem. Okay, that is fusiform cambium, or increasing the girth or thickness. Okay, produce secondary tissue. This is produce secondary tissue. The main result is shows thickness. Okay, thickness of organs or stems. Okay, these fusiform initial cells divide into two daughter cells. Okay, two daughter cells, and one of which become either xylem mother cell or phloem mother cell. Xylem mother cell or phloem mother cell, depending on its position. Okay, depending it's on position. Okay, gradually these cells are divided into lot of divisions. Okay, the secondary xylem laid down towards center and secondary phloem towards periphery zone of the stem. Periphery zone of the stem. Okay, lower side means central side xylem, forming a xylem, and peripheral side phloem. Here this is phloem, lower side, center lower side xylem. This is fasciform or fascicular cambium reactions. Okay, next. Next, secondary xylem uh, is nothing but wood. Okay, constitute the bulk of woody plants. Okay, take care of transport of water, mineral salts, mechanical support. Okay, mechanical support and storage of food in the wood parenchyma. Okay, wood parenchyma made up of xylem vessels, tracheids, xylem fibers. Okay, the xylem fibers are performed mechanical functions because of their cells completely filled with hard material, that is lignin or lignified walls. Okay, lignified walls. Xylem parenchyma in the wood commonly stores of food material like starch and fats, tannins, crystals. Okay, this is the wood xylem. Wood xylem importance. Okay, next, secondary xylem shows hardwood and sapwood. Okay, two types of uh, wood is there, that is diffuse porous wood and ring porous wood. Diffuse porous wood, ring porous wood. Okay, diffuse porous wood means shows small pores. Okay, uniformly throughout the growth rings, shows small pores. Okay, that's why commonly called diffuse porous wood. Okay, second type ring porous wood. In this type of wood shows vessels appear in two ways in two seasons. That is, spring season the vessels are more closely placed. The wood is larger. Then autumn season vessels are more widely spaced. The wood is smaller. Wood is smaller. Okay. Next, uh, sapwood and hardwood. Okay, in trees, after some formation of considerable quantity of secondary wood, two parts, that is sapwood, hardwood. Okay, The outer region of the wood is called sap wood. Okay, young xylem vessels. Shows yellow in color. Sap wood, yellow in color. It helps storage and conduction. This is the sap wood. Okay, this is the information of secondary growth of plants. Next, and hardwood and anomalous secondary growth is there. Next, hardwood. The central dark place is called, that is hardwood. Okay, this is, lesson is separate is there. Tomorrow we will discuss wood anatomy. Okay, now uh, anomalous secondary growth. Okay, anomalous secondary growth. Uh, Anomalous secondary growth in different plants. Okay, anomalous secondary growth in different plants. Okay, different plant means first normal secondary growth. The most of the top root and main lateral roots of the dicotyledon plants shows secondary increase in thickness. Secondary growth is present in monocotyledons roots. Okay. Ah, then abnormal secondary growth. Abnormal. Okay, abnormal secondary growth uh, occur four stages or four reasons. That is abnormal behavior of normal cambium and formation of accessory cambium, formation of intraxillary. Okay, intraxillary phloem and secondary growth in monocots. Okay, this is the reasons abnormal secondary growth. Okay, abnormal secondary growth. Abnormal behavior of normal cambium. Okay, abnormal behavior of normal cambium. In many lions, lions means uh, 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 woods or trees are uh, creeping on the trees. Okay, creeping on the trees. The type of small plants is called lions. Okay, that is lions. Okay, normal cambium in lions, many lions behave abnormal, resulting formation of more amount of secondary phloem. Secondary xylem, more amount of xylem than phloem. For example, bignonia. Okay, bignonia. Bignonia plants shows abnormal secondary growth. See here, this is not bignonia. Mm, yes, this is a bignonia. Okay, bignonia shows anomalous secondary growth. Okay. uh, in each rings, okay, some environmental effect on the plants shows abnormal conditions. Okay, more amount of secondary phloem than secondary xylem. Okay, bignonia TS transfer section shows epidermis, cork, epidermis, cork cambium, cortex, endodermis, and pericycle fiber patches. Next, starch sheath, phloem wedges, secondary phloem, Then the next part is cambial strips. Okay, the cambial strips is very important abnormal changes in bignonia. Okay, secondary phloem. Then after form a secondary xylem. Okay, after that parenchymatous medullary ray. Okay, the central portion occupied by fifth or medulla. Central portion is fifth or medulla. Okay, here for cambial rings cases to function after some time. Okay, the accessory ring of cambia are formed secondarily. Okay, which continues the secondary growth. Okay, the abnormal cambia generally originate the cortex. Abnormal cambium generally form a cortex region. Okay, epidermis. Then next portion is cortex. Okay, here each cambium ring produce xylem and conjunctive tissue inside flowing parenchyma outside. Inside form a xylem. Here, inside form a xylem. Outside form a phloem and parenchyma. And form a four wedges. One, two, three, four. Four wedges of strips. Okay, four wedges of strips. This appearance, okay, concentric rings of vascular bundles embedded in the conjunctive tissue. This type of abnormal secondary growth is common in the member of Amaranthacea. Okay, Amaranthacea family. Okay, this is the bignonia. Okay, next bignonia. Okay, bignonia woody climber. Okay, lions and climbers. Okay, this is woody climber means wood nature is very hot. Okay, yet shows anomalous secondary growth and cambial strips. Four cambial strips shows four cambial strips. Okay, thus adaptation to the climbing habit of the plant. Okay, this anomalous secondary growth in bignonia stem described as adaptive anomalous secondary growth. Okay, adaptive anomalous secondary growth. Okay, this is the information of anomalous secondary growth in bignonia. Next, next plant is Lyptodendron Borehavia. Okay, Borehavia. This is the Borehavia anomalous secondary growth figure. Borehavia shows epidermis. Okay, the second layer is hypodermis. First layer is epidermis. Second layer is hypodermis. Next parenchyma. Okay, here not shows strips, four wedges strips. Okay, here vascular bundles are scattered manner. Okay, okay. First layer is epidermis, hypodermis, parenchyma. Okay, epidermis. Second layer hypodermis. Hypodermis made up of collenchyma tissue. Then third layer that is parenchyma. Fourth layer starch sheath. Okay, vascular bundles covered by starch sheath. Then after secondary phloem, form a secondary phloem by cambium. Next, secondary xylem. Okay, secondary xylem. Then conjunctive tissue. Okay, so central portion is occupied by medullary bundles, primary medullary bundles. Okay, all are arranged in ground tissue. All are arranged in ground tissue. Okay, in ground tissue. Okay, here the abnormal second growth found in the different plants. That is, first one is here Borehavia. Okay, Borehavia internal anatomy. Okay, in this type of anatomy, interxillary or included phloem developed by variation in cambial activity during the secondary growth. At the time of secondary growth, plant shows this type of characters. Okay, especially Salvedra, uh, Cumbratum shows. Okay, this is a non-adaptive type of anomalous secondary growth. Okay, vascular bundles are different here. Okay, that is non-adaptive. Secondary bundles are shows strips, four body strips. That is adaptive, adaptive type of anomalous secondary growth. But here non-adaptive, non-adaptive type of secondary growth. This is the Boerhaavia diffusa plant anomalous secondary growth. Okay, generally secondary growth is two types: intrastellar and extrastellar. Intrastellar secondary growth, extrastellar secondary growth. Okay, the secondary growth main key role play by fascicular cambium. Already we discussed fascicular cambium. Okay, next, uh, anatomy of stem. Anatomy of stem. Till now we are discussing about growth, TS, stem TS. Again, different types of plants is there. That is anomalous secondary growth in Acheronthus and uh, different types of Bignonia stem. Okay, Bignonia stem. Bignonia is a common garden climber. Common garden climber to the family is Bignoniaceae. The young stem Bignonia is transverse section shows different outlines. That is epidermis, cortex, endodermis, pericycle, vascular bundles. Hmm? Okay, epidermis, that is upper layer or single layer with cuticle. Okay, the parenchyma is prominent below the ridge and collenchyma. Cortex is the made up of few layers of parenchyma. Pericycle is a sclerenchymatous. Okay, present in the below the ridges as patches. This is the information about Bignonia. Next, Draceena. Draceena, the secondary growth is unusual phenomena among the monocotyledons. Some members of little flora shows abnormal growth, that is Draceena and Yucca. Okay, Agave, Aloe Vera, all these are shows anomalous secondary growth. A cross section through the stem. A cross section through the stems of Draceena. Okay, the relevant reveal the different characters. Okay, primary structure is epidermis, cortex, vascular bundles, or ground tissue. Next, anomalous secondary growth. Okay, epidermis is made up of single layer, compactly placed, cell covered by the thin cuticle cortex. Next, vascular bundles. Okay, the ground tissue. Ground tissue forms the bulk of stem, in which are embedded many vascular bundles. Okay, many vascular bundles are embedded in the central portion of the stem. Okay, and scattered condition. Vascular bundles are scattered in the different manner. Primary vascular bundles oval and generally concentric. Okay, primary vascular bundles are oval shape and concentric. So they are amphi-vessel type. Both vessels is there. That's why commonly called amphi-vessel type with central patch of phloem surrounded by xylem. Some of these vascular bundles are collateral collateral and conjoint. Vascular bundles are open type. Different types of vascular bundles is there. Already we are discussed in last class. Okay. Some vascular bundles are open type. Some vascular bundles are collateral type. Okay. Open and collateral. But here vascular bundles are collateral type. In Draceena. In Draceena. Okay. What is occur in Draceena anomalous growth? Okay, second growth is initiated by the differentiation of cambium in the cortex. Okay, the activity of cambium is unusual. It produces more cells inwards than outwards. More cells inwards than outwards. Cell produced, the cell produced inwards get transformed into parenchymatous. Okay. The cell formed outwards by the cambium are parenchymatous. The constitute the secondary cortex. They are thick-walled. Okay, secondary cortex, thick-walled and lignified. Thick-walled and lignified walls. Okay, this is the information of it is that Draceena. The last one is Dacus carota, anomalous secondary growth. Dacus carota shows epidermis and hypodermis. Okay. Next, vascular bundles. Okay. Here, stored food material in secondary growth. That is vascular. That is primary vascular and diarch. Vasculature and diarch. The secondary growth formed of cambium in the interstitial parenchyma. Okay, this is the beet root anomalous secondary growth. Okay, uh, next one is uh, tylosis. Okay, tylosis already we are discussed, not discussed. Okay, tylosis is a wood property. Okay, tomorrow classes continue with the time tylosis topic. Okay, now remaining part is anatomy of
Sir, what's the page number, sir?
page number 78. Hello?
Okay, sir.
78. Anomalous secondary growth of different stems. Okay, that is Draceena and Darkus carota. Okay, different plants were studied. That is the anomalous secondary growth. Okay, and they are studied dicot and monocot stems, internal anatomy. Okay, last class. Okay, dicot root and monocot root. Cortex is narrow in dicot root, but monocot cortex is very wide. Okay, hello. This is main difference between dicot and monocot root. Next, uh, exodermis is absent in dicot root. Monocot is present. Exodermis is sclerenchymatous. Okay. Next, in dicot root, uh, medulla is very small or absent, but in monocot, medulla is very developed. Okay. The number of xylem and phloem strands in dicot roots 8. Okay, 2 to 5 in some shows 8. But in monocot root, xylem phloem strands are numerous, means number of more than 8. That's why polyarch. Okay, polyarch means number of. Okay, in dicot roots, xylem vessels are generally angular, but here oval, angular, oval. Okay, this is the information of dicot, monocot, root. We have discussed last class. Okay, we have discussed last class. This is the information of secondary growth of different plants and anomalous secondary growth. Okay, Borehavia, Draceena, Yucca, beetroot, or Beta vulgaris. Okay, all these are the information. Okay, what is the definition of secondary growth? An increase in plant growth, increasing plant growth due to vascular cambium or carc. Okay, the main key role play in the secondary growth is carc cambium or vascular cambium. Okay, vascular or carc. Then the gradually these cambium rings form a fascicular cambium. Okay, these fascicular cambium form a different cells. Okay, cell division by cell division divided by two daughter cells. Okay, again these cells are periclinar anticlinar divisions shows then form a thickness. Secondary xylem is laid on central side, means medulla side, center portion, that is xylem side, form a xylem portion. Outside form a phloem portion. Increase the phloem portion means increase the amount of phloem outside and increase the amount of xylem inside by fascicular cambium. This is the main difference in secondary growth. Okay, what is the result of the secondary growth in plants? Increase in the girth. Hello, increase in the girth or thickness.
Diameter.
Hello, students.
Yes, sir.
Hello.
Yes, sir. Yes, sir.
Yes, sir.
Ah, okay. This is the all cells are made up of this type of cells. All these are hello. Hello, students.
Yes, sir.
Yes, sir.
Which type all these cells are? Which type of cell or cell system? Okay. All are forgetting. Meristems. Meristem systems. Okay.
Yes, sir.
Meristems systems. Okay. Last class all are forgetting internal structure of dicot stems. That is last topic. Internal structures of stems, roots, primary and secondary structures of roots and stems over and secondary growth and abnormal growth is over. The next topic is plant taxonomy. Okay, students.
Yes,
Next
sir.
topic is plant taxonomy. Okay. One topic is new topic is there here. That is wood structure. Wood
Yes, sir.
structure and properties. Wood structure and properties topic is there here. Okay, this type of wood is developed by uh, vascular bundles. Okay, and given different types of quality wood. Okay, that is hardwood, sap wood, color wood. Okay, commercial wood. Okay, all types of wood is there in our surrounding nature. Okay, tomorrow we will discuss different commercial woods and wood quality and tylosis. Okay, tylosis also tomorrow we will discuss. Okay, today class is over, students. Hello.
Sir, again, some important questions you told us, sir, you will give.
Okay, I will give you today.
When, sir? Next Monday, from next Tuesday, exams will be starting, sir.
Okay, 22. next week on which date?
Yes, sir.
Which date?
From 22 exams are starting, sir.
22 onwards?
Yeah, sir.
Okay. I will send you. I will send you
preparing
today.
So we can
Okay.
I will.
That's why you forward the last class also, sir. Actually, we are preparing for second semester exams right now.
Okay. I will give you today. Okay. No worries.
Yeah. Your number is added in the group, sir. I have sent you the message also for the important questions. You didn't reply.
Okay. But this type of okay, how many people are there in group? Hello?
So many are there, sir. 15, 20 members. 16.
Oh, okay. This is group only meant for botany class or all subjects?
No, sir. Botany, zoology, chemistry.
Okay, all subjects are there here.
Yes,
all
sir.
faculties.
Only as BZC students, sir.
What?
BZC students,
BZC
sir.
students, sir. Botany, zoology, and chemistry.
Yes, sir.
Okay. Form only single group. Or separate group is there?
No, sir.
Botany separate, zoology separate.
No, sir. Only one group. BZC group, sir.
BZC group. Okay, send me text once. I will send. Okay.
Okay, sir.
My number is there, na?
Yes, sir.
9989807071.
Yes, sir.
Okay, I will send you. Right?
Okay, sir.
Okay. Bye.